Alright, going to look at some organelles here, specifically the nucleolus and the ribosomes, which we'll come to in a second. First we're going to look at the way that you can actually go about studying these things. And before we get to organelles, I'm going to give you another example of it. What you need is a piece of equipment called a centrifuge. Now if you look at it from the top, this is basically just something where you can put two test tubes at either end of a kind of holder, and it's going to spin round. That's basically all it is. Okay. If you're looking at it from the side, here's my test tubes. You know, you'd have something to kind of hold them in place like that, and this would be fixed onto a motor that would just spin round. All right. Now, if I was doing this for something like blood, I could put a couple of samples of blood in. Some of these have multiple tubes in there. I've just drawn two. And this would be enclosed in a box because this is going to rotate several thousand times a minute. So this is pretty quick revolutions. It works on the basis of mass. So what it will do is any heavier things, in our example here we've got blood. The heavier parts of the blood, the red blood cells, will get forced towards the bottom of the tube as it's spinning around. It's a bit like a roundabout or a fairground ride where things get forced out as it spins, okay? And I could leave that in there for a few minutes, and then if I took it out, took the tube out, what you'd end up with is all of the red blood cells, the big heavier parts, would be sitting at the bottom, and on top of it you'd see a kind of yellowy coloured liquid, the plasma, various bits and bobs in there. This stuff is sometimes referred to as the sediments, and this stuff is a nice name. It's called the supernatant. Usually that's not the bit you're after, but if we wanted to look at the red blood cells, for example, this is a way to isolate red blood cells from our sample. Now, we can do the same thing with organelles of cells. Now, we'd have to rotate this for longer and at very high speeds because we're talking about things which have a much smaller mass, but it would work. So if we centrifuge things, the first things that would come off would be the heaviest organelles, which, if you can think about it, would be actually the nucleus. Okay, so if you put a load of cells in here, you know, you'd usually mash them up, you'd have some kind of liquid in here, isotonic, meaning it's the same concentration of salts and sugars as the cells, perhaps a buffer solution. So this is a really common thing to see when you talk about cells. It's called an isotonic buffer. A buffer keeps the pH the same. So if there are any changes, if the cells start pumping out acid or taking it in for some reason, the buffer sort of absorbs it. Okay, and you have natural buffers in your body. Isotonic keeps it the same concentration, and it stops the cells bursting. So you'd put it in a liquid that's isotonic buffer, pop it in your centrifuge, whack it on, go and have a cup of tea if you want. When you come back, you'd find that the nuclei had all settled towards the bottom of the tube, and you could get rid of your supernatant, or you could take your nuclei out. Now, different organelles obviously have different sizes and different masses, and so you could keep, you know, the longer you did this, the further and further you'd get down. The smallest of which would be, oops, I can spell it properly, the ribosomes, and these are the really tiny little dots, you know, we see, and obviously we get them as well. Those are free ribosomes, but we get them on the rough endoplasmic reticulum, where they're embedded in a membrane. So if we wanted to study ribosomes, we'd have to centrifuge our organelles for a while. So, what are the ribosomes doing? Well, they're manufactured in the nucleolus, or synthesized in the nucleus, this blob within a blob. You know, here's our cell, here's our nucleus, and we've got the nucleolus sitting in there. So its main job, as far as we're concerned, is manufacturing ribosomes. Ribosomes are made of a combination of something called ribosomal RNA and a combination of proteins. So there's lots of proteins, multiple proteins involved here, you know, sort of in tens of proteins at least, sort of 10, 20, 30 odd proteins, whatever it is. And these things are all combined together and they make this little organelle called a ribosome. It actually consists of two parts. I don't know why I'm changing colours to red, but I am. That's a rough drawing of it, that's kind of close enough. And they're called the small and the large subunits. Okay, and they fit together. And our ribosomes, which are going to be involved in protein synthesis, they're going to be involved in reading off the messenger RNA and converting it into the transfer RNA is going to come in here. You'll do all that when you do protein synthesis, but it kind of clips around the messenger RNA, if you like, that sits through the middle, and it's got these spaces for the transfer RNA to fit in. But you come across, it looks like a skull now, doesn't it? It's made of 2 subunits. Now, one thing to be aware of, in prokaryotic cells, the ribosomes are slightly smaller. They're actually called 70S ribosomes, and in eukaryotic cells, the ribosomes are slightly larger, and they're called 80S. Don't worry what the S stands for. If you want to look it up, you can, but it's not important to us. It's actually to do with the centrifuging. All you need to remember is that these ones are slightly larger. Although, interestingly, if you look in mitochondria and chloroplasts, which, if you remember, do have their own DNA and they do have their own ribosomes in there, the ribosomes in the mitochondrial chloroplasts are small ones. And remember this idea that we talked about, that mitochondrial chloroplasts potentially were separate organisms that were taken into larger cells and have this relationship where the mitochondrial chloroplasts benefit and so the other cells. But again, this fact that they have the smaller ribosomes probably supports that theory quite well. But even among prokaryotes, so the bacteria and the archaebacteria and the eukaryotes, these ribosomes are pretty similar. They're pretty much the same. They're not massively different. And again, it points to this idea of probable common ancestry if you go back far enough. So ribosomes, which are involved in protein synthesis, can be found on the rough endoplasmic reticulum, can also be found floating free, and the difference between the prokaryotic and eukaryotic is just this size.